Hi, I'm Kyle Procopio, and for these last 10 weeks, I've been working with my mentor, Byron Rusnak, in the Rotor Lab, and together we've been able to examine the effects of abscisic acid, brassinosteroids, and jasmonic acid on sepal initiation. Okay, that work? No? Okay, I'll just use the computer. Okay, so in the Rotor Lab, we study robustness, which is the consistency and reproducibility of organ shape and size under the constant strain of environmental factors. Specifically, we're looking at the model system Arabidopsis lyrata, or not lyrata, Thaliana, in sepals, which is the most outer wall of the flower, and we can see it as the flower bud there. Robustness is extremely important, not just for plants, but all organisms, as it can lead to many developmental defects. We look at the gene DARMI1, also known as VOS2, which stands for variable organ shape and size. It has many contributions to phenotype, which includes plant height, root length, but what we're most interested in is it delays sepal initiation, which contributes to both sepal count and sepal surface area. So what is VOS2? Let's take a look at the wild type of buds and their sepals. So here's our bud, and if we take a look at the sepals, all of them are roughly the same size, and they equal to about 90 degrees for the full circle. VOS2 is a little different, however, if we look at the bottom chart. And as you can see, not all of them have 4 sepals, which is a big difference. Some have 5. So this doesn't exactly equal to the 90 degrees that we're looking for. VOS2 is known to be influenced by different hormones, such as auxin and cytokinin, which are both very important plant phytohormones. Auxin has a very weak response in VOS2 plants, where cytokinin has a very strong response, as we can see in these two graphs here. So if it's influenced by auxin cytokinin, why can't it be influenced by other hormones? And that's what my mentor set out to do. He performed a single cell RNA sequencing test, and he was able to find supporting evidence that abscisic acid, ABA, and brassinosteroids, BR, are both upregulated in VOS2 plants, where jasmonic acid, JA, is decreased. So, knowing this, abscisic acid and brassinosteroids are increased in VOS2 plants. So if we were to disrupt their signaling with either a chemical inhibitor or a disrupting mutant to its signaling, this would result in a rescue of the phenotype, resulting in a wild type lookalike. Opposite to that, if we were to start with a wild type plant and increase the signaling of ABA and BR, we would result in a VOS2 plant. Totally contrasting this is jasmonic acid. Now, in VOS2 plants, it's downregulated, but if we were to upregulate this, we would be able to potentially rescue the plant.
It just didn't get plugged in.
Okay. Okay, and then opposite to that, if we were to start with a wild type and downregulate jasmonic acid signaling, we could result in a VOS2 plant. So how do we go about doing this? We start with an Arabidopsis plant, cut off its inflorescence, and place it on a media plate, which has all the necessary nutrients that is needed for it to survive. If there's any chemical treatments that we're taking a look at in the experiment, they're also added. I've listed which ones we're taking a look at here. And from there, we dissect these inflorescence even further in my last picture here. And I've shown you exactly how many buds there are, because it's very hard to see, and they're very small. So small that they're at one of the youngest stages, stage 3, which is when the primordius or sepal primordius begin to arise. And this allows us to take a look at confocal imaging. 2 to 4 days later, the plant is able to grow, and we stain our samples with propydium iodide, which allows us to visualize cell walls and give us these wonderful pictures here. So what are our treatments? For ABA, we're taking a look at the chemical inhibitor, ABA antagonist 1, and we're looking at a few mutants as well, ABA deficient 1-5 and ABA insensitive 1-1 and 2-1. For our brassinosteroid treatments, we're taking a look at one of the most bioactive brassinosteroids that there are, brassinolide, also known as BL. Its chemical inhibitor, brassinazole, BZ. And we're taking another look at 3 mutants, brassinazole resistance 1-1D, which is its dominant form of the gene, brassinosteroid insensitive 1, and deetylated 2. Let's talk results. To remind you, our wild type is on the left here, and all sepals have roughly the same size, but in the VOS2 plant, we can see that the most outer sepal, which in all of these pictures will always be on the top, is way more prominent than the wild type plant. Additionally to that, VOS2 sepals are not always at the perfect 90 degrees that wild type buds have. When we apply 1 micromolar of brassinazole, we're able to see that in VOS2 plants, it totally rescues the VOS2 plant here. So we acquire that nice 90-degree angle, and all sepal sizes are roughly the same size. The outer sepal is no longer extremely prominent. However, we did find a dose-dependent change. When looking at 5 micromolars, we see that this is obviously not the case, as when looking at the VOS2 5 micromolar, we acquired some 5 sepal buds, as well as a variance in angles. And I recognize that our sample sizes are a little low, and this is something that we want to continue looking at in the future, but this is a good lead. So if brassinazole affects plants, why can't brassinolide, since brassinazole affects brassinolide biosynthesis? So here we found that there's an increase of buds that have 5 sepals instead of 4 in VOS2 plants when we add 40 nanomolar of BL. And this is just an example of one of our buds that have 5 sepals. For our jasmonic acid treatments, and this is something we do in a future study, as I was not able to acquire enough time to complete this, we're looking at the jasmonate methyl jasmonate, a chemical inhibitor, JARN1, and 3 mutants once again, carnitine insensitive 1-22, OPDA reductase 3, and jasmonate resistant 1. So in our looking to the future, we've taken a look at a lot of fluorescent proteins. And what we can do with this is we can visualize where certain hormones are within the plant itself. So there's already a lot of fluorescent proteins on auxin and cytokinin that are relevant to the inflorescence and sepals. So if we look at the graphs here, red is cell walls in all of these pictures. And in the left picture, we're looking at auxin here. That's where auxin is prevalent. And if we outline these, we can see that they're mainly on the tips of all the sepals, where in contrast, if we look at cytokinin, and it's a little harder to see on the picture on the left. So auxin is on the tips of the sepals, where cytokinin is on the outside ring of the buds. So what can we do with this in my project is that if we were to find a brassinosteroid reporter, we can hopefully find some crosstalk between auxin and cytokinin if these hormones are relatively in the same spots or not in the same spots. Additionally, if we were to find evidence that jasmonic acid is being influenced by VOS2, we would do the exact same for jasmonic acid. And since there's already good evidence in the literature that abscisic acid is being influenced in VOS2 plants, we've already begun crossing our fluorescent protein 6X ABRE GFP with our VOS2 plants. Lastly, in our future directions, we would really like to make an updated model of VOS2 and all of its relationships with these hormones, as well as other items such as sepal initiation and organ development. There's already been a few models, but we would really like to update these as we now know that auxin and cytokinin are involved and brassinosteroids as well. And I'd like to thank my mentor, Byron Rusnak, once again, as well as Julia Kong and Mingyuan Zhu for all their former work on the subject. And I'd like to thank the Rotor Lab and all of you as well. Thank you.
Great talk, Kyle. Is this on? Yeah. I know you really like phytohormones, so I'm wondering, this is just a basic question, but what is the coolest thing about how hormone signaling works that you learned this summer that you didn't know prior to coming to this internship?
Yeah, so I've mentioned crosstalk within this presentation, and it's a really interesting concept that hormones interact with other hormones and their response to each other affect other responses. And there's so many different effects that these hormones have, and we can never really know how they're going to interact with each other. So that's really cool, especially with auxin and cytokinin, since there's a lot of crosstalk between them. Thank you.
Great talk. I really, I loved all like the images that you had in there. It was just a great way to be able to visualize. I was just wondering, you know, you perform these experiments on an agar plate.
Yes.
Is there a reason why you did that instead of doing it like in planta, or is it just like logistically it would be too much to try to do that?
Yeah, so when working with the confocal microscope, there's only limited space, and having it on a very thin surface like a media plate is very beneficial for that. Yeah.
Hi, Kyle.
Mhm.
So during your results, you mentioned that a 5 micromolar dosage of brasinozole did not rescue the VOS2 mutant. It actually exacerbated. Is correct?
Mhm.
So how would you test further if it is dose dependent or not moving forward?
Yeah, so there's a few ways. And first of all, I'd like to recognize the sample sizes again were not the greatest, but we would like to still continue the study. There's a few things in that. We can study the mutants, which directly impact brassinosteroid signaling. And we can use live imaging instead of what I just did, which was regular imaging, which is a day-to-day analysis of our confocal microscopy. So we test it for every 12 hours, take pictures, and that could really show us what exactly is happening with these hormones.
We have